Hello everyone, welcome back to Sedu Setsavi YouTube channel. So we are back with our Food Safety Officer Exam 1000 MCQ series. So in today's MCQ discussion, we'll discuss the question number from 11 to 20, the second part of microbiology discussion. So without any further delay, let's move on to the first question of the day. The first question is, who is considered to be the father of modern microbiology? A choice, Louis Pasteur. B, Robert Koch. C, Paul Ehrlich. D, Anton van Leeuwenhoek. E, Edward Jenner. So let us understand a few facts about the scientist given in the option. The first one is Louis Pasteur. So you know that Louis Pasteur is considered to be the father of microbiology. Father of microbiology is Louis Pasteur. Apart from the designation father of microbiology, Louis Pasteur was, you need to remember a couple of points. He was the one who introduced the germ cell theory. So germ cell theory was introduced by Louis Pasteur. Also, the fermentation principle was introduced by Louis Pasteur. Fermentation principle. So these are the important points you need to remember with respect to each scientist. Now, Louis Pasteur was the one who introduced the vaccines for cholera vaccine, anthrax vaccine, then rabies vaccine. So you can remember CAR vaccine, cholera vaccine, anthrax vaccine, and rabies vaccine. So these are the points you need to remember with Louis Pasteur. Now coming to Robert Koch. So Robert Koch is the father of bacteriology. Father of bacteriology is Robert Koch. And Robert Koch is considered to be the father of modern microbiology. Is also considered to be the father of modern microbiology. So if the question is who is the father of modern microbiology, the answer is Robert Koch. And if the question is just the father of microbiology, the answer is Louis Pasteur. Now, one more point you can remember about Robert Koch is that he was the one who discovered the anthrax bacillus, then tubercle bacillus, also the Vibrio cholerae. Okay, so these are the contributions of Louis Pasteur and Robert Koch. Now coming to Paul Ehrlich. Paul Ehrlich is considered to be the father of chemotherapy. Paul Ehrlich is considered to be the father of chemotherapy. Also, one more point you need to remember, Paul Ehrlich was the one who discovered the acid fast stain. Acid fast stain. So you know that acid fast stain, which is used for staining the mycobacterium. So that was initially discovered by Paul Ehrlich, and later on it was modified by Zeal and Nelson. We'll come to that actually. So these are the 2 points, important points you need to remember about Paul Ehrlich. Now coming to Anton van Leeuwenhoek, he is considered to be the father of light microscopy. Now coming to Edward Jenner, the most important point, he developed the first vaccine for smallpox. First vaccine for smallpox. So these are the important points you need to remember with the scientists given in the choice. So the answer for this question, father of modern microbiology, is Robert Koch. Louis Pasteur is father of microbiology. Paul Ehrlich, father of chemotherapy. Anton van Leeuwenhoek, microscopy. Edward Jenner, smallpox vaccine. Okay. Now coming to the second question of the day. Which of the following mechanism is involved in the chemical method of sterilization by phenol? A choice, cross-linking and alkylating. B, protein precipitation and cell membrane damage. C, oxidizing agent. D, all of the above. So in the previous discussion, we have told that sterilization can be divided into two methods. Method of sterilization of mainly two types. One is the physical method of sterilization, and the second one is chemical method of sterilization. Now, in our previous video, we have given a detailed explanation for all the important physical methods. Now let's look into the chemical method of sterilization. Now, as the name indicates that in chemical method of sterilization, we use chemicals like alcohol, aldehydes could be used, phenol could be used, halogen compounds could be used, oxidizing agents could be used, even gas sterilization involves a chemical, we'll come into that. Salts like silver salts could be used, surfactants could be used, even dyes could be used as a method of sterilizing by chemical means. So these are the examples for chemical method of sterilization. Now, in the case of alcohol, you know that ethanol ethanol and isopropyl alcohol, IPA, abbreviated as IPA, isopropyl alcohol. So these are commonly used as an alcohol for sterilization. Now the mechanism of sterilizing here, it is denaturation of the protein, denaturation of bacterial proteins. Denaturation of bacterial proteins are the mechanism by which alcohol does sterilization. Now in aldehyde, you can need to remember two examples. One is formaldehyde, that is HCHO, then glutaraldehyde. So aldehydes, formaldehyde and glutaraldehydes are the one which is used. Now here the mechanism is cross-linking. In aldehyde, the mechanism of sterilization is cross-linking and alkylating property. Alkylation. Now phenol is a the examples include cresol, okay, then Dettol is a modified phenol and all that. Here the mechanism is protein precipitation. Protein precipitation plus cell membrane damage. Cell membrane damage. Now coming to halogens, like chlorines are used as a for sterilization, and the main mechanism, because chlorine and all that are oxidizing agents. So halogens, the mechanism of sterilizing is basically oxidizing property. Okay. Now we have separate category of various other oxidizing agents. The main examples are hydrogen peroxide, H2O2. Peracetic acid is also an oxidizing agent. Peracetic acid. Even the technique plasma sterilization is a kind of plasma sterilization basically use H2O2. So it is an oxidizing method. The mechanism of action is oxidizing agent. So as the name indicates, these are oxidizing. The mechanism how it sterilizes that it causes oxidization. Okay, so that is how it will sterilize. Now coming to gas sterilization, the chemical used is ethylene oxide. Ethylene oxide is the chemical used in the gaseous sterilization, and the mechanism is alkylating agent. They are alkylating agent. Alkylating agent. Now coming to silver salts, salts like silver salts, the main method of sterilization is by protein coagulation. They are protein coagulants. They coagulate the proteins. Now surfactant, the mechanism of action is loss of membrane permeability. Loss of membrane permeability plus protein denaturation. Protein denatured. When it comes to dyes, various dyes are used, occurred in dyes, and they basically inhibit the synthesis of peptidoglycan component of cell wall. Okay, so these are the mechanism by which all these chemicals causes sterilization. Okay, so alcohol, aldehyde, phenol, halogens, oxidizing agents, gas sterilization, silver salt, surfactant, dyes. Now coming back to our question, which of the following mechanism is involved in the chemical method of sterilization by phenol? Phenol. So the answer is protein precipitation, phenol, P and P, protein precipitation and cell membrane damage. So the correct answer is B choice. Okay, let's move on to the third question of the day. Chloroxylenol is present in A. Dettol, B. Savalon, C. Both A and B, D. Tincture iodide. So you all know that Dettol, Savalon, they are basically antiseptics, right? Now the composition you need to know in Dettol. So this is Dettol. In Dettol, the active ingredient is the active component or the active ingredient is chloroxylenol. So the active ingredient present in Dettol is chloroxylenol, and the concentration is 4.8 percentage weight by volume. That is the concentration used in Dettol. Now, apart from this chloroxylenol, the inactive components or the inactive ingredients present in Dettol, you need to remember one is pine oil. Pine oil. The example is alpha terpineol. The main component is alpha terpineol. Apart from pine oil, isopropyl alcohol. I told you the abbreviation is IPA, 12 percentage. Then castor oil soap. Castor oil soap is also an inactive ingredient. Then caustic soda solution. Caustic soda means NaOH solution, caustic soda solution. Then caramel, also water. So these are the inactive ingredient, and the main active ingredient is nothing but chloroxylenol, and the concentration you can remember 4.8% weight by volume. Now Savalon is also an antiseptic, and in Savalon the active or the component is chlorohexidine. chlorohexidine. The main component is chlorohexidine. Even in the bottle, you can see chlorohexidine is given. Okay, chlorohexidine plus cetrimide. Cetrimide is a surfactant. Okay, and here the concentration of chlorohexidine is 0.3 percentage weight by volume, and cetrimide is 3 percentage weight by volume. Okay, so that concentration also you can remember. Okay, so the component of Zavalone is chlorohexidine and cetrimide. Okay, now coming back to your question, chloroxylenol. So the answer for this question is Dettol, a choice. Okay, chloroxylone. Savalones, it is chlorohexidine and cetrimide. Whereas what is tincture iodine? Tincture iodine is also an antiseptic. It is nothing but 2 to 3 percentage of iodine solution. Tincture iodine is nothing but 2 to 3 percentage of iodine solution in water alcohol mixture. In water alcohol mixture. So the percentage is 2 to 3 percentage iodine solution in water alcohol mixture. They are basically used as an antiseptic. So that is called as tincture iodine. So here the answer is Dettol. Okay. Now coming back to the fourth question of the day. Which of the following is a test for disinfectant? A. Phenol coefficient test, Riedel-Walker test, Chick-Martin test, Kelsey-Sykes test, all of the above. So first thing you need to know, the effectiveness of a disinfectant or antiseptics, they are mainly determined by four types. Okay, so to test a disinfectant or to test the effectiveness of a disinfectant, we have four types of test. So that is one is the Riedel-Walker test. The second one is called as the modified Riedel-Walker test. Riedel-Walker test is also called as phenol coefficient test, and the modified Riedel-Walker test is called as Chick-Martin test. The third one is called as Kelsey and Sykes test, which is also known as capacity test. The fourth one is called as Kelsey and Moore test, which is also called as in-use test. Now let us understand a few facts about these four tests. So the first one I told you, Riedel-Walker test. Riedel-Walker test is also called as phenol coefficient test. Okay, so here mainly the microorganism taken is Salmonella typhi. Okay, so a broth of Salmonella typhi is taken. Now we'll test the effect of disinfectant in this. Okay, now for that, for a control, we will take phenol, because you know that phenol will kill Salmonella typhi. Now, to test the efficacy of a new disinfectant, you will compare the effect with the phenol, already existing disinfectant, standard disinfectant, and you will compare the effect of this disinfectant with the phenol in killing Salmonella typhi. So, the equation for the phenol coefficient test is highest dilution of disinfectant needed to kill Salmonella typhi broth divided by highest dilution of phenol needed to kill Salmonella typhi broth. So, this is the equation which is used to find the coefficient, the phenol coefficient. Okay. Now, if you get the value, if this coefficient value is 1, if you get the value 1, what does it mean? If the value is 1 means your test disinfectant is as effective as phenol. Okay. So, if you get a value of 1 from this equation, that means the test disinfectant is as effective as phenol. Now, if you get a value which is more than 1, that means the test disinfectant is more effective than phenol. So, what does it mean? That means higher the phenol coefficient value, higher the phenol coefficient value, what does it mean? It means the disinfectant is more effective. More effective is the disinfectant. More effective is the disinfectant. So the take home message is that if the value of phenol coefficient is higher value, then the effectiveness also will be higher. Okay, so that is all about Riedel-Walker test. Now, one disadvantage of this test is that it does not give an idea how effective the disinfectant in the presence of organic matter. So the disadvantage is that it does not indicate how effective the disinfectant in the presence of organic matter. So that is one disadvantage. Organic matter means in the presence of yeast, in the presence of fecus, in the presence of soil. We don't know how effective this disinfectant is. Now, to address this, the modified test is there. So that is called as the modified Riedel-Walker test, and that modified Riedel-Walker test is called as Chick-Martin test. Now, the advantage of Chick-Martin test or modified test is that the test will be conducted in the presence of organic matter. Okay, presence of organic matter like fecus, soil, or yeast. So that is one advantage. So this is a modified form of Riedel-Walker test called as Chick-Martin test. Okay. Now the third test is called as Kelsey test, also called as the capacity test. Now here, this test will measure the capacity of the disinfectant. It will measure the capacity of disinfectant to retain its activity in repeated use. So what is the capacity of the disinfectant on repeated use? Okay, it will measure the capacity of a disinfectant on repeated use. So that is a test that is called as Kelsey-Sykes test. Coming to, and here in Kelsey-Sykes test, the microorganism what will be used is Staphylococcus aureus, E. coli, and Pseudomonas aeruginosa. Okay, so that is all about Kelsey and Sykes test, also called as the capacity test. It measures the capacity of a disinfectant to retain its activity on repeated use. Okay, now the fourth one, Kelsey and Morer test, is also called as in-use test. So this is used to test the contamination of disinfectant. Okay, so contamination of disinfectant is carried out by a test called as Kelsey and Morer test. It is also called as in-use test. It is also called as reality testing. Reality testing, the real test. Okay, the contamination test. So that is all about Kelsey-Morer test. Now coming back to your question, which was a test for disinfectant. So all these phenol coefficient tests, the other name is Riedel-Workel test, the modified test called Chick-Martin test, Kelsey-Sykes test, all are basically the test used for disinfectant. So the correct answer is E choice. Now coming back to the fifth question of the day, which of the following is an example of symbol stain? A. Methylene blue. B. India ink. C. Silver stain. D. Gram stain. E. Acid fast stain. So first thing we need to know the types of staining. So basically we can divide the stain into four categories. The first one is called as the symbol stain. The second one is called as the negative stain. The third stain is called as the impregnation stain. And the fourth one is called as the differentiation stain. Now what is the difference between these? Now you look at the name of the first one, that is symbol stain. So here only one color would be there. Okay, so if you have all different bacteria, it will give same color to all bacteria in the smear. Okay, so only it gives only one color to all bacteria in the smear. And the commonly used dyes are methylene blue. If you use methylene blue, the color will be blue. If you use basic fusion, the color will be red. So basically it will give only one color. Okay, so the examples of the dyes used in symbol stains are methylene blue and basic fusion. Sometimes in question they will ask which is an example for a symbol stain. So either methylene blue or basic fusion. Now coming to the second one, that is negative stain. So negative stain, here the background will be stained. Here the background gets stained. That is the difference here. Okay, so the background gets stained, so therefore the sample or the bacteria which is unstained will stand out in that contrast. Okay, so the background will be stained, the bacteria will not be stained. Since the background is stained, the bacteria, we can see the bacteria, okay, on that contrast. Okay, so the dyes mostly used in negative stain is India ink and nigrosin. Okay, so the dyes used are India ink and nigrosin. Now coming to impregnation stain, so so normally the bacterial structures or the cells that are too thin, if the bacterial structures or the bacterial cells are too thin, then we cannot see under light microscope. Therefore, they are thickened by you impregnating these structures by silver. You impregnate the surface with the silver so that then it will be easy to visualize. Okay, so that stain is called as impregnation. So mainly it is used for thin structures, for example, flagella. Flagella is a very thin structure. So you will impregnate, you will add silver to that so that it will be thickened and you can visualize that. So that is called as impregnation stain. And common example is silver stain. And the commonly used structures are, one is flagella, also the spirochetes. Spirochetes means the treponema group. A treponema pallidi and treponema group can be stained using silver stain or impregnation stain. So remember flagella and spirochetes. Now the fourth one, that is the most important one, the differential stain. Here, as the name indicates, different colors will be there. Okay, so it has different colors. Okay, so this is the most commonly employed stain called as the differential stain. It imparts different colors to different bacteria and different structures. And the commonly used examples are gram stain, acid fast stain, and Albert stain. So gram stain, acid fast stain, Albert stain are examples for differential stain. Methylene blue is an example for symbol stain. India ink is an example for negative stain, and silver stain is an example for impregnation stain. Now, one more thing: the most out of this four staining, the most important, as I already told, it is gram staining. Okay, so it is developed by the scientist Christian Gram. So gram staining, the most employed staining, is developed by the scientist called as Christian Gram. It is the most commonly employed stain. Now you need to remember this. You can remember this mnemonic: CVIAS. CV of an IAS person. Okay, CVIAS. What does that mean? CV means crystal violet. Okay, so here crystal violet is very mostly used, and that is the primary dye or primary stain. Okay, now I means what? I means iodine. Okay, CVIAS. I means iodine, and iodine is an example for a mordant. Mordant means basically they help to fix the dye on the slide. Okay, for example, to fix the crystal violet on the slide, the mordant is a fixative that will help the crystal violet to stick onto the slides. So that is called as mordant, and iodine is a mordant. Now the third one, is called as a decolorizer, and the decolorizer which is used is alcohol. That is, so A stands for alcohol. Okay, CVIAS, alcohol is commonly used, and the third step, so that is act as a decolorizer. Instead of alcohol, you can also use acetone. Now in CVIAS, S stands for saffronin, and saffronin is a secondary dye or a secondary stain. Okay, apart from saffronin, you can also use a dilute carbol fusion or neutral red. Normally saffronin is used. Okay, so you can remember CVIAS, crystal violet, that is the first one, crystal violet. Then iodine is the mordant. Then alcohol is the decolorizer. Then saffronin is the secondary dye. Okay, so that is the point you need to remember. Now one more thing you can remember, whenever you use a gram staining, we are talking about gram staining, right? So mainly we can use to differentiate gram positive and gram negative bacteria. Okay, so after the primary stain, once you use crystal violet, the gram positive will also get the violet color. Gram negative will also get the violet color. Okay, so after the first step, gram positive bacteria will get the violet color, gram negative bacteria will also get the violet color. Now you will use the mordant iodine, so that crystal violet will get fixed. Now in the third step, what will happen? Decolorizer. When you use alcohol, gram positive will remain violet only. Violet means purple color. Okay, no change for gram positive. But gram negative bacteria, what happens? The violet color in gram negative, the violet color will be removed. Okay, so that will become colorless. So by the third step, gram positive will still remain violet color, but a gram negative bacteria will become colorless. Now in the final step, that is the fourth step, what you do, you are adding a secondary dye. So gram positive always be violet, no change for that. Now gram negative, which is colorless, now it will take the color of saffronin. Saffronin means red color or pink color. So gram negative will become red or pink. So that is how gram staining can differentiate gram positive bacteria and gram negative bacteria. Okay, so at the end of the stain, gram positive will look like violet in color and gram negative bacteria will take the red or pink color. Okay, so these are the steps involved, and this is the color change in between gram positive and gram negative. So you can remember the mnemonic CVIAS, crystal violet, iodine, alcohol, saffron. Now coming back to your question, which of the following is an example for a simple stain? The answer is what? Yes, methylene blue is a simple stain. Okay, now let's move on to the next question of the day. In gram staining, already we have discussed, in gram staining, the mordant is, I told you, CVIAS. CV is the primary dye. Iodine is the mordant. Alcohol is the decolorizer. The saffronin is the secondary dye. So which is the mordant? Yes, the answer is iodine. Now coming back to the next question of the day. Gram positive and gram negative organisms are stained dash and dash respectively after the entire gram staining procedure. A. Purple and purple. B. Pink and pink. C. Pink and purple. D. Purple and pink. So I already told you, in the case of gram staining, you need to remember this mnemonic CVIAS. Okay, so CV means crystal violet. After staining with that, you will get the violet color. Iodine is the mordant. Then alcohol is the decolorizer, and saffronin is the secondary color. So this is the primary color, and alcohol is the decolorizer, and saffronin is the secondary color. Okay, so in the case of gram positive, I told you, and gram negative, both will take the violet color. That means violet or purple color. So after the first step, both gram positive and gram negative will get violet or purple color. Now second method is iodine. It's fine, it will be fixing. Now decolorizer. Now after the decolorization, what will happen? Gram positive will be still violet or purple, no change for that. However, gram negative will become colorless. Okay, gram negative will become colorless. Now after the last step, the secondary dye color, gram positive will be violet or purple. However, the gram negative will take the saffron in color, which is nothing but red or pink. Okay, so that means at the end of the thing, the gram positive will be violet or purple, and the gram negative will be red or pink. Okay, so what will be the answer? Gram positive and gram negative, the color respectively they asked. So the answer will be gram positive will be still purple in color, whereas the gram negative will be red or pink. So the correct answer for this question is D choice. Let's move on to the next question of the day. In acid fast state, okay, the primary and secondary dyes dash and dash respectively. Okay, so earlier we have told about gram staining. Differential stain contains gram stain, acid fast stain, and the Albert stain. Now, acid fast stain, the first thing I already asked you to remember, it is discovered by Paul Ehrlich. Paul Ehrlich. Now, this acid fast stain is, although it was discovered by Paul Ehrlich, it was modified by Zeal and Nelson. It is modified by Zeal and Nelson. So that is why it is called as Zeal-Nelson staining or ZN stain. Okay. Now, this acid fast stain is mainly used to stain the mycobacterium. It is used to stain the mycobacterium. Mycobacterium, you know that Mycobacterium tuberculosis is there. Mycobacterium tuberculosis, Mycobacterium leprae is there. So that is the main use of acid fast stain. It is used to stain mycobacterium. Now here also in the gram staining, I told you CVIAS, right? Now here you can remember this mnemonic CHAM, CHAM. Okay, so in acid fast stain, you need to remember the mnemonic CHAM, CHAM. What is C stands for? C stands for carbonyl fusion. That is the primary dye. What is H? H is the heat. So in gram staining, it was iodine. So here H, that means heat, is the mordant. Heat is the mordant. Now A stands for acid, which is the decolorizer. Here acid is the decolorizer. Instead of acid, you can also use alcohol. Now what does it M stands for? M is methylene blue, or you can use malachite green. So that is the secondary dye which is used. Okay. So here the mnemonic CHAM, you can remember, primary color is carbonyl fusion and secondary color is either methylene blue or malachite green. Now one more point you can remember. I told you the here the decolorizer is acid, right? So acid, the normal commonly used acid is H2SO4, that is sulfuric acid. Okay, and in the case of for Mycobacterium tuberculosis, the acid percentage used is 20% sulfuric acid is used, 20% H2SO4 used. Whereas in the case of Mycobacterium leprae, it is 5% sulfuric acid. 5% sulfuric acid. So these are the important points you need to remember with the acid fast staining, mainly used for detecting mycobacterium. It is also called as Zeal-Nelson stain, CHAM. Okay, so the question was primary and secondary. CHAM, carbonyl fusion, is the primary dye, and methylene blue is the second. So carbonyl fusion and methylene blue is the answer. So the correct answer for this question is A choice. Now coming to the next question, silver impregnation staining is used to demonstrate. I told you simple stain is there, then negative stain is there, then impregnation stain we use silver salt, right? Silver salt we use. I told you mainly it is used for thin structures. It is used for thin structures. Once used impregnated silver, it will become thick so that it will be easy to visualize. Now it is commonly used for thin structures like flagella. Flagella. Also I told you spirochetes. Spirochetes like a treponema group of microorganism, treponema. Okay, so these are the two common thin structures like flagella and spirochetes can be visualized or stained using silver impregnation staining. So the answer for this question is B choice. Mycobacterium is stained by which method? Mycobacterium is stained by which method? Yes, acid fast staining. Okay. Now let's move on to the last question. What percentage of sulfuric acid for Mycobacterium leprae in acid fast staining? So I told you in acid fast staining we will be using decolorizer, right? Decolorizer will be used. Acid is normally used, and which acid? Sulfuric acid, H2SO4. So I told you in acid fast staining normally we will use it for Mycobacterium tuberculosis, TB. Also it is used for Mycobacterium leprae, leprosy causing organism. So you can remember in the case of Mycobacterium TB, the percentage of sulfuric acid is 20%. You can remember T20. Okay, T20. Mycobacterium tuberculosis, 20%. Okay, whereas Mycobacterium leprae, the percentage of sulfuric acid is 5%. You can remember LEPRA, leprae. Okay, just you can LEPRE, you can remember five alphabets or something. So 5% is the amount of sulfuric acid used. Okay, so the question was Mycobacterium leprae. So the answer for this question is 5%. Okay, so hope you understood this 10 MCQ discussions from microbiology. Keep on watching. Thank you so much.